Good afternoon, everyone, and welcome to our webinar titled The Standardisation of Australia Treatment Pathways for Cholangiocarcinoma. Today's webinar is a very proud collaboration between the Cholangiocarcinoma Foundation Australia and Omico. Before I introduce our speakers, a few housekeeping items. So today's webinar will be recorded and we will send out a link to the recording after the webinar. We ask you to please revisit the content and to share this content far and wide with as many of you as your colleagues as possible. All microphones, except for those of the speakers, will be muted, and we encourage you to ask any questions in the Q&A section at any time. We'll hold the questions for the discussion at the end, and for any questions that we can't answer today, I'll endeavour to get some answers to you by email. So my name is Dr. Natalie Rickers, and I'm very honoured to be hosting today's webinar. I've spent the last 20 years of my career working in the field of genetics and genomics. My most recent position was working for a US genetic testing company called Invitae as the business development manager for the Asia-Pac region. Very coincidentally, during my time at Invitae, many of my colleagues in the US were working closely with the Cholangiocarcinoma Foundation in the US and with companies like AstraZeneca. It was at that time that my mum walked into my kitchen one afternoon looking extremely jaundiced. A week later, she received a diagnosis of stage 4 cholangiocarcinoma. I now volunteer my time with the Cholangiocarcinoma Foundation in Australia. And before we dive into the webinar, I've got two very distinct memories that I wanted to share with you. The first memory was on the day of my mum's diagnosis. Like many good scientists, I came home and the first thing I did was dive into as many research papers as I could possibly find. What I read in those research papers was truly harrowing. I realised that the odds were stacked against my mum. The 5-year survival rate for cholangiocarcinoma remains less than 5%. And the founder of our foundation, Steve Holmes, is one of the very few people in Australia to have ever survived this cancer. And Steve actually lost his brother to the same cancer a few years before his own diagnosis. Second memory that I wanted to share with you was from one of my mum's monthly oncology appointments. The registrar that day had left her screen open as we walked into the room, and in her Google search bar, she had typed, "What is cholangiocarcinoma?" Now, please know that this is not intended in any way to be a negative comment on the standard of care that my mum received. It was second to none, it was world class. But to me at that point, it really highlighted that with such a rare cancer, there are so many challenges. There's an extra additional level of complication, a lack of resources, a lack of training, and I guess, a lack of treatments, for want of a better word. Like many caregivers who've lost loved ones, I honour my mum's memory with the work that I do with the foundation, and I couldn't be more honoured to have three very distinguished and esteemed clinicians and scientists joining us today to help drive education and awareness for cholangiocarcinoma in Australia. I hope that this will truly help drive change for the many patients that we talk to on a daily basis in our foundation. Today's speakers are Professor David Thomas, Professor John Zalcberg, and Professor Juan Valle. Professor David Thomas is the inaugural director of the Centre for Molecular Oncology at the University of New South Wales. He was previously head of the Genomic Cancer Medicine Laboratory at the Garvan Institute of Medical Research. As a clinician scientist, his focus is on the application of genomic technologies to the understanding and management of cancer. He established the not-for-profit company Omico to lead a national precision medicine program for patients with rare and early onset cancers. Omico is changing the way that we fight cancer by accelerating access to precision oncology. As a national, independent, not-for-profit organisation, Omico is uniting Australia's world-class institutes, researchers, clinicians, industry partners, government, like never before. By leveraging a nationwide network of expertise and resources, Omico aims to improve outcomes for all Australian cancer patients. With fast-tracked molecular and genomic screening, biomarker-led trial setup, and patient enrolment from the entire nation, Omico is truly accelerating access to next-generation treatments and preventative strategies. Our second speaker, Professor John Zalcberg, is a medical oncologist at Alfred Health Until recently, he was the head of cancer research program in the School of Public Health and Preventative Medicine at Monash University. Professor Zalcberg's clinical areas of interest include gastrointestinal cancer, the diagnosis and management of patients with gastrointestinal tumours, and quality of care. He's published over 380 peer-reviewed journals. Professor Zalcberg has held the Tony Charlton Chair in Cancer Research at the Alfred Hospital until 2023. Prior to taking up that position, he was the Director of Cancer Medicine at the Peter Mac in Melbourne for 17 years and was the Chief Medical Officer for 5 years. He's received many awards, which include a Medal of the Order of Australia, the Cancer Achievement Award from the Medical Oncology Group of Australia, and the Tom Reeve Award for Outstanding Contributions to Cancer Care from COSA. Our final speaker, Professor Juan Valle, was most recently a professor at the University of Manchester in the UK, at the Institute of Cancer Sciences, and an honorary consultant specialising in hepatopancreatobiliary cancers and neuroendocrine tumours at the Department of Medical Oncology at the Christie NHS Foundation. His primary focus is on clinical research leading to practice-changing clinical trials with links to translational laboratories. Professor Valle has international leadership roles in societies such as ESMO, as well as working in partnership with patient charities. He now serves as the Chief Medical Officer at the Cholangiocarcinoma Foundation in the US, leading research initiatives and providing strategic guidance in developing innovative treatment approaches for cholangiocarcinoma. Professor Valle is based in Colorado, and due to the time differences, is unable to join us in person today and has kindly recorded his presentation for us. At this time, I'm going to hand over the floor to Professor David Thomas. Professor Thomas, over to you.
This is a very an important topic for oncology right now, because cholangiocarcinoma, in my experience over the past really maybe out to 10 years, but certainly in most cases much less than that, has gone from a disease where we had chemotherapies, which were quite toxic and largely ineffective, to having a whole range, a raft of targeted therapies, and more on the way, which are proving to be effective. And I guess that would be my focus on communication today, was to give you an idea about our experience with cholangiocarcinoma patients being referred onto the program, and what we've learned from that. And I'll try to share with you some live data. If I can, I will share with you some live data about what we have learned. So let me just stop sharing for a moment and go back to my. So in total, Omico's had 839 patients referred with cholangiocarcinoma. Of those, 724, or 86%, have consented to taking part in the program. And that's an important point. I'm somewhat surprised sometimes to see patients who are referred by our, the clinical community, but actually for some reason or other don't end up taking part in the study. And that might be something that we could improve if we're to reach the maximum potential of this technology. Of those that are consent, that is of 724, 553, or 86%, completed successful sequencing. Now, there are many reasons why we don't get successful sequencing. Amongst those are too little tumor in the tissue, on pathology review, the specimen itself is too small. There's a whole lot of reasons why that percentage contributes. But one thing it's important to say, it's not particularly different for cholangio than it is for the cohort as a whole. Of the 553 who were sequenced successfully, 453, or 82%, had a matched recommendation for a therapy. And of the 453, 75, or about 15%, 1 in 6 patients, went on to receive a matched therapy, and some patients have received more than one matched therapy. Of that 75 patients, 51 received treatment on clinical trials, which partly reflects the availability of many of the most effective therapies over the period in which we're recruiting patients, something for a discussion perhaps later. 1 patient received treatment on the PBS, and 12 patients privately funded, 8 received compassionate access, and in 5 patients we don't really know we really don't know what mechanism they use to be able to access the recommended matched therapy. Now, some general points about MoST and CaSP. As you know, our first program started in 2016 called MoST, and it completed in December of 23. And our current program is called CaSP, and it's worth making a couple of points about the difference between the two. So in the MoST program, we were trying to screen in last line because we wanted to make sure that patients who took part were past conventional therapy, and we wanted to make sure that there'd be as much actionability as possible, which was probably in retrospect not correct, because about half the patients didn't go on to receive a subsequent therapy. And so in CaSP, we've specifically decided that as soon as a patient has incurable disease, that we're open to performing sequencing. And that's particularly true for diseases like pancreatic cancer and cholangiocarcinoma, where the 6 weeks taken to get a result can be too long for some patients. Now, in respect of that 6 weeks, which is our expedited or urgent category, when you say it's urgent, that's what the category that we put it into, 2 weeks of that is spent in actually getting the blocks into the path lab. The path lab then receiving the sample takes 2 weeks to get the result back to us, and it takes us under 5 days typically to turn around the molecular report. So another point here is, if you've got a patient that's urgent, everything you can do to encourage the pathology lab and the patients to consent will actually shorten that turnaround time even further, which is obviously something that we all want. Now, on to the good stuff. So what I'd like to do now is share with you our, I'll go to my desktop hopefully, and you'll be able to see the raw data portal that we have for the patients that we've enrolled, including their molecular profiles. Can you see my screen? Please tell me that you can, if you can.
Yeah, David.
Okay. Can you see this now?
Yep, we can see it.
Yeah, we haven't seen it before now, David, but we can see it now.
Okay, great. So this is the data portal that we use internally when we're trying to look at data quickly. So what you see here is that we have in total 14,530 patients in this data set. We're currently at 20,000 patients. So the numbers that I was showing you earlier was a more up-to-date section. But this is of the 14,530 patients on whom we have some reasonable follow-up. If I select on biliary cancers, then I end up with 606 patients. I'm sorry, my cat is trying to crawl on top of my keyboard. Of the 606 patients with biliary tract cancers, you can see the majority have the histology cholangiocarcinoma. And I want to take you down here to where we see the mutation profile. So you see in the left panel in the middle, the genes, and on the right panel, the specific mutations that we're interested in when we make recommendations. What I'm going to do now is I'm going to select the genes and mutations which today I regard as having at least phase 2 evidence of improved survival. So the first is KRAS, and you'll see of the 143 with KRAS, 67 are G12D, and another 7 are G12C, which means more than 50% of the KRAS mutant cholangiocarcinoma currently has a therapeutic or a therapeutic class which would be regarded as druggable and where there's emerging evidence for response. Then we go to IDH1, FGFR2, MTAP, BRAF, with V600E turning up somewhere around 8 out of the 25 patients, BRCA2, ERBB2, IDH2, and in total, the number of patients out of the 606, let me just go up to sort of illustrate this point, is 377 of the 606 patients that I'm reporting out on have one of these targets. These are not targets where the responses have not been observed. We know that from the emerging, and it's rapidly emerging through abstract form for the most part, but we know that these mutations are things that we would strongly recommend trying to get access to treatments on. Down the bottom, you'll see the tumor mutation burden profile, which isn't captured by those individual genes, and you'll see that it's 3% of the cohort, 2.5 or 3% of the cohort have a high tumor mutation burden, which adds a little bit to that actionability on top of what I've just described. So look, I might stop there because I'm very keen that if people want, they can ask me some questions towards the end and I can pull up and parse out the data if I'm able to on request. But the message I'm trying to send is this is a disease where very personally over the past 6 or 7 years, we started off having very little, and it's astonishingly changed compared to what it was back in the middle 2010s. So I think it's very exciting, and I'm really looking forward to some of this data maturing. I'll stop there.
David, what was the, can you just repeat, what was the percentage of patients with a mutation, a targetable mutation overall?
377 of 606, so about, I would say 60%.
Yeah.
Yeah. And some of it's, you know, not things you'd expect. I didn't go down into the minutiae at the long end of it. There's some rare things in there like BRCA1 we turn up. There's some RAD51s. There's certainly BRCA2. As you know, John, we see BRCA2 mutations and germline mutations in patients with cholangiocarcinoma, not to mention the usual spectrum of ERB2. And that's not taking into account ADC targets. Those are genomically mutated ERB2. So there are, I think there's enormous potential here. And of course, the antibody drug conjugates are emerging, which will only help fill in this with time.
All right, thank you, David. Apologies to everyone. There's no better time to lose my internet connection than right in the middle of a webinar. So thank you, David, for jumping in there and saving the day. I did have wonderful introductions for all of our 3 speakers. I'm pretty sure that you would all know David and John extremely well. So we're going to hand over to Professor John Zalcberg next. Thank you, John.
Thanks, Natalie. Okay, so this is quite different focus, but I guess just illustrates some of the work going on in Australia around the management of cholangiocarcinoma or biliary cancer. Go the next slide, please. So the background of this is that we've had a sort of focus on trying to make sure that we measure quality of care, and the reason for measuring it is because we know there's lots of variation. I'll show you why that exists in a moment. So for a complex disease, the diagnosis is complex, the treatment is complex. You've just heard David describe a whole lot of different mutations, which leads to different treatment. Some people are not having mutation testing done, etc. So there's going to be variation. And to understand the extent of that variation, we do need to understand it because it's the only way we'll see where that variation is appropriate or not. So sometimes it's very appropriate that someone with a BRCA mutation is obviously going to be treated differently to a patient with metastatic disease and a mutation in the RAS gene. So that's an example of appropriate variation. It's not even variation, it's just appropriate therapy, but treatment differs between those examples. But other situations where there may be variation that is not, it's not clear why it exists or why it occurs. And so the only way to understand that is to actually measure what's going on. And this slide is really meant to say that we need to, because there's variation, let's measure it, let's measure what's going on. When we measure it, we can benchmark institutions and see where that variation is common to all or not. And then when we benchmark, we can understand how to, we can at least start to understand how we might improve quality of care. And as treatment evolves and these treatments that David has talked about come on stream, some of them, sometimes they come on, they're adopted quickly, sometimes they're not. So by benchmarking these practices across institutions, we can see where there's room for improvement. Next slide, please. The important thing about quality of care, of course, is that it's delivered by the clinical community. It obviously involves patients and consumers, but requires significant clinical engagement. And I think it's a really important part of trying to define optimal care is that you engage clinicians, whether they're medical doctors or dietitians or physiotherapists or social workers, you engage clinicians in understanding what the problem is and engage clinicians in understanding how to go about improving things. And so engagement is a really fundamental part of any attempt to understand quality of care. And it means that when we define optimal care, we define that with the clinical community. We don't define it for them. We don't tell them what's optimal, but the clinical community actually defines what's optimal care, and then can become part of the way of moving forward in terms of resolving that. Next slide, please. So how do we decide what is high quality care? And there are various ways of doing that. Obviously, you can go to the literature, but the literature is often distilled into guidelines. They can be international guidelines. They can be national guidelines. There can be a clinical consensus. All of these processes need to involve consumers in those processes. But at the end of the day, that allows you to say, what is evidence-based best practice that determines optimal care? So evidence-based best practice, because that's what's optimal. And then if we know what's optimal, then how do we measure that? So if optimal care, for example, is you should have a mutation test done in a newly diagnosed patient with metastatic cholangiocarcinoma, if we all agree as a clinical patient community that optimal care involves molecular testing, then whether it's occurring or not is relatively straightforward, right? Number of patients diagnosed with metastatic disease, was mutation testing done, yes or no? The answer to that question might be, let's say it's done in 70% of patients. The other 30% doesn't mean something wrong's been done. It means that there may be explanations for it, but at least that gives us a chance to understand why that variation exists. And if you've got one institution where mutation testing is done in 90% and another where mutation testing is done in 30%, that allows both of them to reflect on what they're doing, how they're doing it, and whether they can improve. In Australia, there are no national guidelines for the management of biliary cancer. There was no formal clinical consensus of what represent optimal care. And so this project that I'm going to describe in a bit more detail in a moment was about trying to redress that issue and see if we could come up with some management guidelines that were relevant to the Australian population. Next slide. And just to finish off this, I really described this when you collect data around what's going on. So that's this, and the tool we use is a clinical quality registry. You've got clean data, you look at where there are outliers, you feed that back to organizations, and you end up with improved care. Next slide, please. That's the theory. So we needed to have some treatment pathways in order to be able to say, well, how do we think treatment is best provided in Australia? And so we had a small group. This was David Goldstein is on the line, myself, and Charles Pilgrim. I'm not sure if Charles is on the line or not, but really set about to try and understand if we could develop a set of treatment pathways that would inform a registry in the future when it's finally established. So as I say, it's really the drive of David and Charles, with me hassling them at various times, but them hassling me to try and get to this point. So I'll show you some of the work we've been doing. Next slide, please. So we formed a steering committee. You can see the members of the steering committee here. It's representative of the broader Australian community. Kirsten Barton is at the end here, helped me develop these slides, is a project manager at the Clinical Trial Centre in Sydney. Licia Greenhill was part of the led the upper gastrointestinal cancer registry at Monash. Melanie Regan, I won't go through all of these names, but just the last three here. And Melanie works at PanCare in Melbourne and is responsible for developing a set of optimal care pathways with Cancer Australia for biliary cancer, and that work is well underway. Next slide, please. We've got as you'd expect, this work did require some funding, and we went about and sought funding from a number of organisations, including PanCare. They're listed there. I should say that one of them was from a patient family donor. I've only just got permission to put them on this slide when I give this talk in the future. But essentially, that's called Ken's Quest, Finding a Cure for Cholangiocarcinoma. So that will be another. I don't have a logo for Ken, but Ken unfortunately passed away with biliary cancer. But his family have raised a substantial amount of money to help this work. Next slide, please. So the first thing that we did is we developed a list of all the international guidelines that were published in the literature at the time. And since we started this work, there were 2 other guidelines that came out, but we've tried to incorporate some of their recommendations. So we took the guidelines that existed. We formed a number of expert working groups, and this was a little bit ad hoc. We tried to invite appropriate representatives of all the various craft groups that particularly clinicians that manage biliary cancer, but it wasn't meant to be these weren't meant to be the only experts, but nevertheless they were people with expertise in this area. So you might imagine that the surgeons, of course, were hepatobiliary surgeons, there were gastroenterologists, there were pathologists, there were interventional radiologists attended various of these expert working groups. We also held focus groups, and I'll talk about that in a bit more in a moment, but then we quickly realized there was a lot of controversy around who needs to be stented, when do they need to be stented, how should they be stented. In the example that Natalie talked about, a mum who was jaundiced, and so clearly a stent is an important part of management, but and we also had a focus group around biopsy, who to biopsy. Well, everyone mostly is biopsied, but how to do that in a way that was feasible and appropriate with the overall plans of management and liver transplantation. We plan to do a real-world practice survey. So the previous expert working groups that I mentioned were really people with expertise, and they were asked to look at these international guidelines that I mentioned and tell us what they agreed with and what they disagreed with. But the real-world survey is going to be much broader population assessment of individual cases, which illustrates some of these points, and we'll see, I don't think any doubt, we'll see the extent of variation that occurs in management. And then we will try, and we've started the process of doing a modified Delphi of actually working out what are the appropriate quality indicators by which we can measure quality of care in this condition. Next slide, please. So the International Guidelines Survey, just to reiterate, there were 7 of them. There were 175 statements in all. We distributed these to these multidisciplinary experts that were part of these groups that I mentioned that met around Australia. And they were asked to agree with these statements, refine them, disagree, or should say there was insufficient evidence. And then what we did, so that was a survey that went out beforehand. And then what we did is we got the results back. We had 75 statements for which there was sort of lack of consensus, and we discussed a number of those in these working groups that I mentioned earlier. So they were held in Queensland, Adelaide, Melbourne, and Sydney. Next slide, please. These are the 7 guidelines that were available at the time we started this work, so towards the end of 23. But in fact, as I say, there are a couple more now that have been published since that time. A number of these go back some time, though. If you look at the dates here, the American guidelines, 2014, and no updates really since that time. So that's a decade or more old. Where else? The European guidelines were more current. They were 2022. And the Japanese guidelines are a bit more current. So you can see that they varied over time and there was some overlap between them. Next slide, please. This one's a bit busy. I might let's go to the next one. So these are the next one. This is, I think the supportive care one is the easiest one. Let's go to the next one. And next one, sorry. Yeah, they're all the same, but I just want, it's a bit easier. No, just go back. Sorry, yeah, this one. This is the role of radiotherapy. There are a number of statements around radiotherapy, and you can see There was a number of statements for which there was agreement, but there was also a number which were thought to be rejected, or there was insufficient evidence. So this is just one of the topics, but you can see that clearly there's lots of consideration around whether those guidelines apply now. Some of them because of time, as I say, if they're a decade old, things have changed, but some of them because there's either evidence that we disagree with or evidence that's not fully mature, and so there will be different views expressed about these various recommendations. But the intention of all this work is to report where there was a consensus and where there was disagreement in order to inform the next steps. Next slide, please. This is the supportive care one. Again, you can see not many supportive care ones, unfortunately. Remember, we didn't create these guidelines. We were simply looking at the existing guidelines and trying to see where there was consensus in the Australian environment. Next slide, please. Excuse me. So these were the expert working groups. As I say, 79 attendees across the country, and you can see in that list a range of people with different expertise, all meeting together to try and understand, how we might reach a consensus around optimal care. Next slide, please. As a result of all this, we've refined 35 of the statements for which there was not lack of agreement. 7 were recommended for refinement with the future working group. There were 12 statements which were retained with no modification. 17 recommendations we didn't think were appropriate, and those 3 topics that I mentioned selected for further discussion or refinement in the focus groups. Next slide, please. And the focus groups, again, were really just structured focus discussions around these 3 areas. You can see that under topics, you can see there was, and I mentioned these before, stent management, biopsy and liver transplantation. The focus groups were held on one afternoon on a Friday. It was a long day for those of us involved in all of them. We had 26 people from various specialties. Remember, they're very distinct topics. So we had 26 experts and developed a range of additional recommendations. I think one of the things that changed out of this is the importance of liver transplantation and a focus on having sort of criteria for when liver transplant's appropriate or not. Next slide, please. So the real world, practice survey is in development. We've got we're going to use a number of cases. We've got half a dozen cases in all. We've nearly finished developing them. They're partly based on real patients and partly based on things that will illustrate the key points and the potential for variation. And I think that these will be important examples of where there is variation and where there needs to be further work in terms of trying to develop a consensus, if not evidence-based practice. Next slide, please. So just with the manuscript for the expert discussions is being developed. The focus group manuscript is well in development. The modified Delphi is underway, and funding permitting, we'd hope to start a registry that routinely measures quality of care in this condition in 2026. I think that's the last slide. Yeah. Thanks, Natalie. Sorry I went on a bit too long, but...
You're welcome. Thank you so much, John. Wonderful to see the amazing work that you guys have been doing. And like I said, I really hope this goes a long way to improving lives for patients with cholangiocarcinoma in Australia. As we queue up the next presentation, I wanted to introduce our final speaker. So Professor Juan Valle was recently a professor at the University of Manchester at the Institute of Sciences and an honorary consultant specialising in hepatopancreatobiliary cancers. at the Christie NHS Foundation Trust in Manchester. Professor Valle has international leadership roles in societies such as ESMO, as well as working in partnership with various patient charities. He now serves as the Chief Medical Officer for the Cholangiocarcinoma Foundation in the US, leading a lot of research initiatives and providing strategic guidance for development of treatment approaches for cholangio. Professor Valle is actually based in Colorado, and obviously due to the time difference was unable to join us in person today, but he's kindly recorded his presentation for us. So we're going to play the presentation. Just a reminder, if you have any questions, please add them to the chat and we'll play them at the end. Thank you.
Hello, good evening. I'd like to thank Omico and CCF Australia, specifically Natalie and Steve, for the kind invitation to speak at this evening's webinar. Sorry I cannot join you because of the time difference. I'm based in Boulder, Colorado, and it's my pleasure to be alongside esteemed speakers. My focus is going to be on discussing treatments beyond cisplatin, gemcitabine, and immunotherapy in patients with advanced biliary tract cancer. These are my disclosures. So let's jump right in. The introduction of immunotherapy, either durvalumab or pembrolizumab, has represented a paradigm shift for the management of patients with advanced biliary tract cancer. These are the Kaplan-Meier curves of the two pivotal studies, the TOPAZ-1 study as well as Keynote 966. Demonstrating an improvement in overall survival, not initially, but after a period of 6 months or so, with divergence of the curves. Quickly highlighting that one of the differences between the two studies is the use of maintenance gemcitabine chemotherapy in the Keynote 966 study. These studies have been updated in terms of overall survival. You can see that the survival benefit is maintained and in fact increases over time, particularly in the TOPAZ-1 study. The question is who benefits most from the chemotherapy, particularly as we are now seeing about 1 in 4 patients being longer term survivors. So who makes up the tail to this curve? And this is something that we've looked at we've asked in a number of different cancers. In an attempt to try and answer that question, Kabir Modi and colleagues published in JCO Precision Oncology a study looking for an immune-related biomarker. You can see from this paper on the left that they identified the biomarkers that we know are associated with a targeted therapy. So that is data that is consistent with previously published data. But on the right, you can see a number of immune cells in the panel, specifically trying to identify an immune-related signature. And what they were able to find is that there were some elements of an immune signature in gallbladder cancer as well as cluster 1. You can see here an increase in B cells and plasma cells, and this represents the most inflamed tumor microenvironment. So I think this work is ongoing for us to identify the patients most likely to benefit from immunotherapy. But this talk is going beyond first line. So let's quickly think about second line chemotherapy. There have been 3 informative studies with second-line chemotherapy, the first with FOLFOX and the second and third with irinotecan-based therapies. In the ABC06 study, we were able to demonstrate a reduction in risk of death by 31% with a hazard ratio 0.69 with FOLFOX after previous treatment with cisplatin and gemcitabine only. At that point, immunotherapy wasn't established in the first-line setting. I think many clinicians were already using FOLFOX. So what this study was able to do was able to have data that would allow us to have an informed discussion with patients regarding the relative merits of chemotherapy, which are modest from use of FOLFOX. In terms of irinotecan, there's 2 contrasting studies here. Both of these studies looked at nanoliposome irinotecan versus 5-FU control group. You can see that one of the studies was positive. The Korean NIFTY study and the German NALIRIC study showed no improvement with the addition of second-line chemotherapy. I think despite this, we know that irinotecan from its use in other GI cancers has some use in biliary tract cancer, and some clinicians do favor that, particularly in patients who already have a lot of platinum-based toxicity. One of the harsh realities is that there is a big drop-off of patients between first and second line and third line in terms of treatment. We know that only approximately 50% of patients receive chemotherapy in the first line setting. 50% of those will go on to second line, and between 20-25% of patients will go on to third line. Again, this Sankey plot was in the era prior to immunotherapy, but I think the key message here is a consistent drop-off of patients as you go through the lines of treatment. Of course, we welcome the introduction of targeted therapies, and we'll talk about that next, but we need to remind ourselves that 50-60% of patients do not harbor a currently actionable molecular target, and this clearly represents an area of unmet need. We have historically considered cholangiocarcinoma based on anatomical subgroups, so intrahepatic cholangiocarcinoma, perihilar, and distal. That classification still holds very relevant in the setting of surgery and local regional disease, so we cannot move away from that completely. But particularly in the setting of advanced disease, we are now considering cholangiocarcinoma as molecular subsets. And you can see here between 2020 and 2023, the amount of information that we're getting to be able to inform the size of the subgroups and the number of subgroups is rapidly increasing. And I suspect that this will continue to change. One of the first treatments that became available was ivosidenib targeting IDH1 mutations. This IDH1 mutated group probably constitutes the largest group within cholangiocarcinoma. The Clarity study was a second-line plus randomized study where patients were randomized 2-to-1 to receive ivosidenib or placebo. In the study, the patients were allowed to cross over on disease progression had they been allocated to placebo. And the Kaplan-Meier that you see includes an adjusted arm to take into account patients who had crossed over, which occurred in around 70% of cases. The top curve is those who receive ivosidenib. Then you have a middle curve of those who received placebo. And then you have a reconstructed arm using the rank-preserving structural failure time model to account for patients who received placebo and modeling as if they had not crossed over. And you can see in that setting, the median overall survival improved from 5.1 months to 10.3 months. There's been a number of FGFR2 targeted therapies. Just to remind you, these are specifically for FGFR2 fusions and rearrangements. This is not a comprehensive list, but just to highlight that there are some consistent themes coming through these different agents. You can see here that there is a large number of patients who have a response according to RECIST evaluation, and even if they don't have a response, there's a large number of patients who have stable disease, and only a minority have disease progression as their best response to treatment. Of these, pemigatinib and futibatinib are now persisting and available for treatments. There are a number of further generation targeted FGFR inhibitors under investigation, including lirafugratinib, the Relay RLY4008 drug, as well as the tinengotinib compound. So this is going to be an area of active investigation still going forward. We know that the impact of immunotherapy in an unselected population, again, is a small incremental step rather than a huge step change. But we know that patients who have MSI high or deficient mismatch repair do have a much higher benefit, as we've seen in a number of other malignancies. In the Keynote 158 study, this was for patients with previously treated non-colorectal cancers. Just pulling out the data of patients with biliary tract cancer and cholangiocarcinoma. There were 22 of those. They're represented in the waterfall plot as a very pale green. The overall response rate was close to 41%. These were durable with a duration of response of 30.6 months. Taking all 22 patients together, the median progression-free survival was 4.2 months, a median overall survival of 19.4 months, and close to a third of patients were alive at 3 years, which is not what we're seeing with an unselected population. Some of the other rarer mutations, BRAF V600E occurs in between 2 and 3% of patients with biliary tract cancer. In this study published in Lancet Oncology now 5 years ago, you can see from the waterfall plot that close to 50% of patients had a partial response from the combination of dabrafenib and trametinib, specifically in BRAF V600E mutated biliary tract cancer. KRAS G12C is an uncommon mutation in biliary tract cancer, where we tend to see more KRAS G12D as well as in pancreatic adenocarcinoma. However, in the Crystal-1 study, which was the phase 2 study of adagrasib, there was a pancreatic cancer cohort as well as other GI cancers. The pancreas data is shown on the left. On the right, you can see the number of patients with biliary tract cancer in the blue bars. And 4 out of 8 patients had a response, so a 50% response rate, including 2 unconfirmed partial responses. So again, the more we do the molecular profiling of these patients, we're going to find some of these rarer molecular subgroups, and increasingly we welcome the introduction of targeted therapy specifically for those patients. HER2 is probably the most hot off the press approval here in the United States. You can see here going clockwise, the HER2 pathway, and then the various ways of targeting the HER2 pathway, including trastuzumab and pertuzumab, which block the dimerization of HER2. You can also see trastuzumab and deruxtecan, antibody drug conjugate. So this now delivers a chemotherapy payload using the HER2 target. We can, at the bottom, use bispecific monoclonal antibodies targeting more than one epitope of HER2, as well as the tyrosine kinase inhibitors, which are targeting the pathway downstream with intracellular tyrosine kinase blockade. And on the left, the combination of a monoclonal antibody as well as a tyrosine kinase inhibitor. So as you can see here, and we've seen this in other cancer types, there are a number of ways of targeting the HER2 pathway. Some of the data, again, you're seeing here waterfall plots, which are very promising with high numbers of patients achieving responses. Another large group of patients having stable disease in the middle, and a relative minority of patients who have disease progression as their best response. What this has shown us is that the treatment options has been gradually accelerating over the past few years. Now, there was a rather large gap in the 2010s when cisplatin gemcitabine was the standard of care before pembrolizumab was approved for MSI high. But certainly over the last 2 to 3 years, you can see here that the number of treatment options that we're seeing is accelerating. And looking at the portfolio of studies which are due to read out, I think this is only likely to accelerate. We've also seen this now integrated in guidelines. So these are the ESMO guidelines. Here in the US, we use NCCN guidelines. I know you'll have local and regional guidelines as well as national guidelines in Australia. I think one of the key take-home messages of the guidelines is that while patients are having their first-line treatment for advanced disease, they need to undergo biomarker testing, molecular profiling. It's called different things. We prefer biomarker testing because that allows you then to have knowledge of the characterization of the biliary tract cancer, specifically with a view to what treatments might be available in second and later lines of treatment. Even if treatments are not readily available, by being approved, licensed, and funded, it will allow us to identify patients who may be suitable for clinical trials or possibly access to expanded access programs. But we also need to take into account that some of these biomarkers are prognostic in terms of survival. So patients having a TP53 mutation, according to this series, have an adverse prognosis, and KRAS mutations is the same. But the opposite is true for patients with FGFR fusions, in whom that fusion appears to be associated with a favorable prognosis. There are a number of series that describe the prevalence of the various biomarker subgroups. This is one of my favorites, largely because the group in Barcelona focused specifically on patients with advanced disease. From the early series, a lot of them focused on patients who had undergone potentially curative surgery, which is a slightly different population to the ones that are often seen in the oncology clinic. This is ranked by the mutations seen in intrahepatic cholangiocarcinoma, IDH1 being the commonest. And you can see as you go down the list the number of different alterations that we have been talking about. You can also see that the profile is very different for patients with extrahepatic cholangiocarcinoma, so that includes perihilar and distal. Often getting tissue there is much more challenging, but we also see HER2 amplifications are more common in that group. One of the key messages that the Barcelona team was also able to show us is that the presence of targetable alteration was prognostically important, as well as, of course, if patients received an on-target treatment. So in this Kaplan-Meier curve, you can see in blue patients who had a biomarker alteration and went on to receive a targeted therapy. They had the best survival with a median OS of 22.6 months. The middle curve is for patients who had a targeted alteration or targetable alteration, but did not receive a targeted therapy, and they had a median overall survival of 19.1 months. And that compares favorably with patients who didn't have a molecular alteration, which are the ones in the pink curve. And I think the observation that some of these targeted therapies are associated with higher responses than we're seeing with either chemotherapy or indeed a combination of chemotherapy and immunotherapy is now raising the question of whether we should be considering those for a neoadjuvant or even an adjuvant approach. Methodologically, that raises a number of questions in terms of single agent versus combination, duration of treatment, and so on. But I think this development is very much welcome as a new research strategy going forward. One of the things that we do know is that with targeted therapies, at some point, patients do develop disease progression, and this appears to be from acquiring resistance. This paper looked at acquisition of resistance in patients with IDH1 mutations. It looked both at AML as well as intrahepatic cholangiocarcinoma. In the ICCA patient, you can see that while they were on ivosidenib, they had a response to treatment, which is shown here. But at the time of disease progression, the patient developed an IDH2 mutation, which seems to indicate an isoform switch from IDH1 to IDH2 as a resistance mechanism. Lipika Goyal and colleagues were able to show in the FGFR setting that resistance can be acquired by the development of new mutations, which can be seen in the tumor biopsy. As you can see here, these are 3 different cases with a baseline scan, the nadir best response scan, and the scan of disease progression. And at the time of progression, you can see the mutations are emerging, and these are also able to be picked up in circulating tumor DNA. And often these are new mutations in ctDNA that were not present as baseline. Often the initial FGFR2 fusion is still picked up at the time of disease progression. So it's important, I think, for us as patients are receiving these targeted therapies to not only do the biomarker testing at baseline, but on disease progression, if there's access to that or within a research program, so that we can really understand what are the mechanisms of resistance which can then be targeted going forward. We know that the presence of co-mutations might also impact on how well patients respond to treatment. In this detailed analysis from patients who took part in the FIGHT 202 study, you can see on the Kaplan-Meier curve on the right, the patients who had a co-alteration with TP53 had a worse prognosis, even though they also received the FGFR targeted therapy. And I think going forward, this is going to lead us to the rational development of combinations with other inhibitors. Either it will be within the pathway or it will be with targeting different pathways, in this case, the EGFR pathway. This is data from Nabil Bardisi's lab in Harvard, and you can see that the combination of infigratinib, FGFR inhibitor, as well as an EGFR inhibitor, the combination of that gives you a much more profound and durable effect. And this may be because it is overcoming some of the escape mechanism from blocking the FGFR2 pathway. So with that, I'd like to quickly summarize. It is an evolving landscape at the moment, particularly in precision systemic therapy for biliary tract cancer. The cornerstones of treatment will remain in terms of chemotherapy, surgery, as well as radiotherapy. Molecular characterization is essential for treatment planning. And here is not just about having one test looking for one molecular alteration. It's have a panel based so that we're looking at a number of different alterations at the same time, but we also get some additional information on what might currently be variants of unknown significance, but in time may be able to make a patient suitable for a clinical trial or help us to understand the impact of co-mutations. We do know that the efficacy of treatment is time limited before resistance develops. And again, this speaks to the importance of testing and retesting, maybe in a research setting initially to understand that resistance mechanism. I think most of us would look forward to being able to identify which of the patients most likely to benefit from immunotherapy, and if patients aren't suitable for that, whether they can then be funneled into additional combination therapies, for example. Future directions, I've talked a lot about understanding resistance, maybe the best strategy for application of these new treatments in earlier stage disease. How do we best select and stratify patients within these clinical trials? And maybe the identification and validation of novel endpoints, particularly with the use of circulating biomarkers. And with that, I'd like to thank you again for your invitation. My apologies for not being there. And just to highlight our forthcoming conference in April in Salt Lake City, where our focus is going to be identifying research priorities in the field of cholangiocarcinoma. Thank you very much for your attention.
All right, thank you so much. That concludes our presentations today. A huge heartfelt thank you to Professor David Thomas, Professor John Zalcberg, and Professor Juan Valle for your incredible insights, your expertise, your input, and your time to the webinar today. As a reminder, we've now got a few minutes for Q&A, so please submit any questions if you have them. I see that we've had a question already in the chat. So this is for you, John, from Lakmali. Thank you, Lakmali. Could you comment on how radiotherapy is used to treat BTC in Australia, and is it consistent across the country?
Yeah, radiotherapy is certainly one of the, we had representation from radiation oncologists in our expert discussions, as I mentioned, that I referred to earlier, like Marley, and certainly I think it's more bespoke. I think radiotherapy has certain roles in, for example, margin-positive disease after surgery. I think there's a role for tumours in the extrahepatic biliary duct, and for cholangiocarcinoma, where surgery may not be possible, but stereotactic radiotherapy has a role. I'm not a radiation oncologist, so I'm sure I haven't covered all the potential scenarios. But so whilst one can talk about situations where radiotherapy is used, whether the same patient would receive radiotherapy in Centre 1 versus Centre 2 versus Centre 3 is a different point, right? And so that's the issue, I guess, that we're trying to understand is, are there examples where it should be used and it's not, or it's not being used and it should? Well, the same thing, but yeah, so you understand what I mean. So I don't think it's standard. I think there's certainly room for these guidelines to provide some insight into the role of radiotherapy. I hope that answers the question.
Thank you, John. Next question. Would it be more practical to have a panel of gene tests specific to ABC than general Omico, which might save time, money, and also might be able to waive consent and do those tests as reflex testing on diagnosis?
Well, foresight would be a wonderful thing. If you were to have chosen a panel 3 years ago, you would have different genes on it today. And in addition to the genes that Juan was pointing out, for which there is excellent evidence, there are more genes which are being identified and targeted today. An obvious example is G12D, and a second example is MTAP, and both of those are relatively common. We also don't capture microsatellite instability on small panels, and we don't capture tumor mutation burden, and both of those may turn out to be quite important over time. I think it's very hard to pin a date where you can say no further development will occur in a disease. And in addition to the things that are more common that I've focused on and have been discussed here, there are also a long tail of individually very rare things which sum to significant options for patients that can only be had with larger panels. So in my view, you need, if you're going to give the best options for your patients, it's best to use as big a panel as is reasonable. And the only thing that you gain, I think, is, for the most part, the turnaround time for some of these panels is less, but you also don't necessarily get all the results that you're after.
Thank you, David. A question for Professor Zalcberg. Do you have an approximate timeline for when the guidelines will be released?
Yeah, it's to say that we've had dates and they keep moving on us. So we think we'll have. Remember, these are not going to be Australian guidelines. These are going to be an assessment of the validity of international guidelines, such as they are, and where we think there's consensus or not. But nevertheless, they will provide some guidance. So I would say, rather than call them a set of guidelines, we would hope that the manuscripts will be out sometime this year. I would hope the first half of the year, but so that's certainly what we're working towards. We are later than we had hoped. We'd hoped to have a manuscript out, at least ready for submission at the end of last year. It just turned out to be an enormous amount of work for a relatively small number of people. I mentioned David in particular and Charles, and the same goes for Pancare. So without trying to continue to make excuses, we recognise it's important to get them out, and we certainly will try and get them out in the first half of the year.
Wonderful. Thank you, John. I don't see any more questions from any of the participants today. I do have a question from one of our patients in our foundation. So last week, she was found to have a BRAF mutation, which is found in approximately 5% of cholangio patients, but it's found in 50% of melanoma patients and approximately 40 to 80% of thyroid cancer patients. Studies have shown significant benefit for patients with BRAF solid tumors, including cholangiocarcinoma, when treated with certain therapies, which we know are approved by the FDA in the US. So the question that we wanted to direct to you today is, can you tell us if there are any kind of ongoing efforts in Australia to move away from offering therapies that are based on the organ affected versus offering therapies based on the mutation?
John, I don't know who wants to tackle that one, but I'll have a brief go and you can hop in. So I think you need to proceed through logical demonstration, don't you? And it would be would have short memories not to remember the 5-year survival for most cancers was 25% pre the sort of routine use of chemotherapy. And chemotherapy still has a 27% objective response rate, as I saw it from Juan's slides alone. That's not to say that chemotherapy is easy or it doesn't have significant side effects or is more effective than targeted therapies. But we need to show that each line of therapy that targeted therapies are reasonable to replace if we're not to do a disservice to our patients. And ultimately, we still have about 30% of our patients, maybe 40%, who don't have one of these good targets, and we need to have something for them as well. Hopefully, all of that will improve with every advance in new therapy. That's my view. John, what you've seen this for? You've seen the
look, I think it's a really good question, and unfortunately, we really don't have access to pan-tumor, drugs that can be used, or targeted drugs that can be used across a range of tumors. There have been attempts by various pharmaceutical companies to apply for what's called tumor-agnostic indications. So the prime example, I guess, that you'll be aware of is microsatellite instability, where the immune checkpoint inhibitors, by and large, are effective in every case, whether you've got endometrial cancer or biliary cancer or colon cancer. If you've got microsatellite unstable disease or abnormalities in microsatellite DNA repair and mechanisms, then the immune checkpoint inhibitors are very effective. And even in that situation, we still don't have pan-tumor approval. And look, it's so there have been attempts by industry to try and change that. The review of the way the PBS operates is complete. The working parties now have been charged with set up by the review panel to try and bring about some of the changes, and maybe this will be one of those. To make it a bit more complicated, though, do you recall that the example we were just talking about, Natalie, was BRAF mutation? Mm-hmm. Now, there were BRAF inhibitors that were used for melanoma, and they were quite effective. Those same inhibitors were then used in BRAF mutations in colon cancer, and on their own, as single agents, they were much less effective. Mm. So you might say, well, just because you've got a certain mutation doesn't mean it will work across a range of different tumor types. Having said that, that particular drug is used in colon cancer, but it's used in combination with other things. So how do we deal with that? I think you can say that the way the government appears to be dealing with it is saying, well, you have to prove that it's effective in every cancer. And of course, biliary cancer is a rare disease. We'll never get these drugs for biliary cancer if that's the case. Yeah. So we do have a problem. But the other thing to do is to think about it, well, the patient, if it's about the patient, which is what it should be about, then the way forward perhaps is to say, well, there's certainly a reasonable hypothesis that drug, that BRAF inhibitor, will work in a patient with a BRAF mutation in biliary cancer. And that's a discussion for the patient and their doctor. Mm-hmm. And there should be at least an opportunity to try. Now, how are we going to pay for that as the community is a struggle with that I don't think we've got a solution for. But so it's a really good question. I wish we had an answer. Mm-hmm. Most of the time, if it was me with that BRAF mutation in a biliary tumor, I'd be saying I need to try and get the drug. And if one could afford it, then that's how you'd get it, because you can buy these drugs, right? But the problem is most people can't afford to pay for them. So it's a very difficult problem.
The objective response rate to combination BRAF and MEK in biliary tract is in excess of 50%. So, you know, this is not a marginally active combination.
Yep. But when will it be approved? When would the PBS ever get a submission, and when would it be approved? I mean, that's the challenge, right? So,
in fact, I think that is the challenge. Now we've got enough of these use cases. I think Omico's got a proposal before the government to try and mainstream this using conventional HTA rather imaginatively. So we're trying to put pressure on the government to think about how to preserve the value for money concept, but at the same time do the right thing by patients with rare cancers in particular. And this is the case.
Yep, I agree.
Yeah, thank you. Thank you for the honest answer. I know it's a very tricky thing to solve. So I think we'll wrap up there. Thank you very much again to our speakers. Thank you to Omico for your very generous collaboration today. It's been wonderful. And thank you to our audience for joining us. If you do have any additional questions, you can email me directly at natalie@cholangio.org, and I'll endeavour to get some answers from the speakers. If we can just move to the final slide, thank you. At the heart of the Cholangiocarcinoma Foundation Australia is what we call the Optimal Patient Response. It's a pioneering initiative that bridges cutting-edge science, clinical expertise, and lived experiences of patients. We have many tools, including the Patient Navigator Journal that's shown here on the screen, mutational translators, and we have a medical professional registry for clinicians who have experience dealing with cholangiocarcinoma. And through all of these tools, we really want to empower patients to engage more effectively with medical professionals to maximize outcomes in their time that they have available. This, we hope, reduces inefficiencies, strengthens collaboration, and delivers better outcomes for everyone. This work has really been transformational, and it helps to, you know, drive patient outcomes. And for us to enable us to continue and scale our efforts, we need your support. So if you found today's discussion valuable, we please invite you to partner with us, to collaborate with us, or help through any other avenues that you may have available. Together, we hope to advance cholangiocarcinoma care and build a sustainable framework that elevates outcomes for patients. Thank you so much again for your time today, and have a wonderful rest of your day. Thank you.